Okay, so in this video I want to talk about a superfusion assay, which is a way in which we can study the endothelium-derived relaxation factor. And we can see, basically, it's a proof that the endothelium is releasing some sort of factor which can then go to the smooth muscle and cause relaxation. Okay, so basically this is what you do. You take a piece of a blood vessel, like so, with an intact endothelium. Okay, so this is an intact endothelium here. Okay, and basically you perfuse this endothelium with a fluid. So you perfuse this blood vessel with a fluid. So you put a fluid moving through this piece of blood vessel, and the fluid is going to contain not acetylcholine. And I'll explain the reason why we can't use acetylcholine in a minute. Instead, what we use is bradykinin. Okay, but for all intents and purposes, bradykinin is going to do the exact same thing to the endothelium as acetylcholine. It'll work for a slightly different receptor. It'll work through B2 receptors rather than M2 receptors. Basically, again, this receptor is coupled to the GQ protein. What's slightly more complicated than that, it's coupled to actually both GQ and GI, but we'll ignore the GI for the basis of this conversation. Okay. So, bradykinin will bind to the B2 receptor on the surface of the endothelial cells, and it will cause the release of nitric oxide from the endothelial cells. So the endothelial cells will produce nitric oxide. Now, some of that will go to the muscle surrounding the endothelial cells in this piece of blood vessel. However, some of it will also go into the bloodstream. So what you do is you collect the fluid coming out, basically. You put it into a tube. So you connect your piece of tissue to a tube. And then what you're going to do is you're going to put this fluid that's coming out through another piece of blood vessel, which has basically had its endothelium removed. Okay, so this is another piece of blood vessel here, which has had its endothelium removed. So it has no endothelium of its own. Okay, and now what's going to happen is this nitric oxide is going to cause relaxation of this tissue. Okay, so you may well want prior to stimulate this other piece of tissue with a potent vasoconstrictor, so that the dilatation effect of nitric oxide will be more obvious. You could constrict it with, for instance, thromboxane A2 would be a common choice. So thromboxane A2 is a very potent vasoconstrictor, and it acts directly on the blood vessels, thromboxane A2. Okay, or indeed you could use acetylcholine acting on the M3 receptors. So what you're basically going to do is you're going to collect the fluid that's coming out of this blood vessel. It will contain nitric oxide, and now the nitric oxide will cause relaxation of the smooth muscle of this blood vessel here. So remember, nitric oxide is the endothelium-derived relaxation factor. And now the reason we would do this experiment with bradykinin rather than acetylcholine is because if we put in acetylcholine, the acetylcholine would come straight through, well, some of it would come straight through, and would then act on the skeletal muscle cell, the smooth muscle cells, and cause contraction, basically. So we use bradykinin instead because smooth muscle cells don't express the receptor for bradykinin, but endothelial cells do, basically. So bradykinin will cause the release of nitric oxide, or endothelium-derived relaxation factor, and if our theory is correct that there is something being released from the endothelium that is causing the contraction of this smooth muscle, then this smooth muscle should relax, basically, and indeed that is what you find.